Hello, today I'm here to talk to you about Emmanuelle Charpentier. Just a warning, my French pronunciation is completely garbage, so I probably said it wrong, so I'm not going to say her name anymore, just for your sake. For her early life, she was really encouraged academically by her parents. Her mother was a psychiatrist and her father was a park manager, and they really pushed her and all her siblings to pursue whatever they thought was interesting. And she had an interest in microbiology from a pretty young age. She attributes that partially to her father explaining the Latin names of many plants to her, but also she doesn't know specifically. There was no one standout moment, but I think that's how it goes for most people. For schooling, for her graduate studies, she investigated segments of bacterial DNA that move around the genome and transfer drug resistance between cells, and she later had a doctorate in microbiology. So she's very focused on microbiology, and she's focusing mostly on the question of how does one specific organism, this one here, this streptococcus one, how does that specific organism have its immunity work? And that's what all of these papers here are going to solve. These are like the greatest hits, the three main ones that I like to talk about because I personally think they're super interesting, even though the titles are kind of extremely boring. Like if I come up to you and say, oh, I read this super fun paper, synthesis of group A streptococcal virulence factors is controlled by a regulatory RNA molecule, you would probably fall asleep. It's just not that interesting of a title, which I'm not judging, it's a scientific paper, they're all pretty boring titles, but it's really interesting what they found. So, they found an unusual RNA that controls the virulence factors in this one specific organism. She'll study this one organism for basically her entire career, and this unusual RNA will come into it. It's a little hint I'm planting in there. I don't know if you'll realize, but it'll come back. And the second paper, that's also an interesting one. This relates to the CRISPR-Cas9 thing. I say thing. It's the big thing she was involved with, along with Jennifer Doudna. Those two together helped make this giant discovery that revolutionized genetics and gene editing for everything, and it's super amazing. And the title is also extremely boring. I'm sorry. I don't want to judge, but come on, look at that last title. Makes you want to fall asleep. Anyway, the second paper found this tracer RNA and this CRISPR RNA and this Cas9. So the tracer RNA right here, this combines with CRISPR RNA. Those two together are basically the sequence that looks for the target DNA. Because what CRISPR-Cas9 does is it cuts a part of the DNA. The CRISPR part, so the tracer RNA and the CRISPR RNA, that's the seeking. The Cas9, it's a protein, that's the cutting. It just chops the DNA. And we need something targeted, of course, because we can't have Cas9 just go willy-nilly, pow, 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 chopping everything up, because that would suck. That would be bad for everything. So we need something to guide it. But there's two steps because you have to have the tracer RNA and the CRISPR RNA combined together, and that's a step. And every step is a waste. So this third paper here, this is like the big paper that changed everything. It's merging CRISPR RNA and tracer RNA into a single guide RNA. That's what the sgRNA is. Wow, we removed an entire step. Now there's one thing to guide and one thing to cut. Better than two things to guide and one thing to cut. You just cut out a step, and that makes it a whole lot easier. And that kind of made it more possible to do human gene editing for genetic therapy and other treatments like that. Super interesting. But yeah, here's a good photo for it. The first one, that's the original one, so you have to combine those two together. The second picture right here, this shows them combined together. After that, she did things. She founded companies. She worked as a professor. She's managing director. She works at different labs. And I really admire that about her. She's definitely done tons of different things. There's a diversity in her interests. And she's really dedicated to what she's done, though. And she's willing to travel. She went all over the world. She started in France, then the US, then Germany, then Austria, and then she's in Germany now. So there's a lot of travel there. Here's a list of some of her awards. There's a ton more. And what is she working on now? Her most recent paper is Survival Strategies of Streptococcus pyogenes in Response to Phage Infections. Again, really boring. But basically, this picture here is describing the functions of different genes, and they were basically trying to find out how does this one organism respond to phage infections, because I know it does, but they don't really know the details, so this is finding out more. How exactly do they use the vesicles? What do they do with them? What is the exact pathway to which it responds? So there's a lot of discovery there, and it's an interesting paper if you get over the boring title. That's from 2021, so it's pretty recent. She's still working with the lab now, so there's still stuff coming out. Here are my citations for photos And then for papers, I hope you enjoyed, and if you do think CRISPR-Cas9 is interesting, I highly recommend the course for spring semester called Genetic Engineering and Editing. It's a great course. You get to edit plants. The teacher brought corn in. It's super fun. So if you want to learn more about this process, I would highly recommend it. From a faceless speaker to the other. Have fun.